our journal club today. So we're very, very, very happy to have Elisa Salido from Valentina Losardo's group at University of Wisconsin-Madison. They want to discuss their latest publication, which was in collaboration with Clint Miller's group from Virginia University, about the role of 9p21.3 as a CAD risk locus that is driving vascular smooth muscle cells to an osteochondrogenic state. So Elsa has a bachelor's in biology from University of Puget Sound in Tacoma, Washington. And then she's currently a fourth year PhD student and recently published this exciting work. So we're very happy to have you today. And I would give the floor to Elsa to tell us more about the research.
Yeah, thank you so much for that lovely introduction. Good morning, everyone, or afternoon, I suppose it would be for many, if not all of you. Uh, like she said, I'm Elsa, and I'll be talking a little bit about our recent publication. So, starting out very broadly, in the Losardo lab, we look at genetic variants linked to complex human disease. And there's a few challenges that are commonly associated with this kind of work. A lot of the variants linked to these diseases are in non-coding regions of the genome, which present their own challenges and tend to be poorly conserved. And they are not necessarily causal. They might just increase risk. So it makes it difficult to parse out precisely what it is that they do. In addition to this, variants in close proximity are often in very strong linkage disequilibrium, so they inherit together. And this makes it difficult to identify which of the variants in a specific region are actually the causal one, or the one that's elevating risk. And finally, of course, different genetic variants as well as different types of disease have tissue and cell type specific effects, which can add kind of another layer of complexity to trying to parse out this sort of issue. And in our lab, we look particularly at coronary artery disease. CAD is a disease that has a pretty strong genetic component. It's around 40 to 60% heritable, and around 300 loci in the human genome have been linked to risk of the disease. Of these, around a quarter are predicted to affect the vascular wall, whereas only 10% are lipid related. But despite this large predicted role for the vascular wall, the roles of specific cell types within that wall remain incompletely resolved with respect to the roles that they're playing in coronary artery disease. So we look at vascular smooth muscle cells, which comprise the majority of the arterial wall and are known to play multiple central roles in the progression of coronary artery disease. Classically, VSMCs are known to undergo a phenotypic switch from contractile to synthetic. And previous work has found that around 70% of cells in the average atherosclerotic lesion are smooth muscle cell derived, so they contribute quite heavily. In addition to the classic contractile to synthetic switch that we see in VSMCs, they can also take on any one of a variety of disease states that have been associated with VSMCs in coronary artery disease, including transdifferentiation, where they just fully turn into other cell types like macrophages or foam cells, and also taking on alternative states where they don't fully transdifferentiate into other cell types, but they take on features of other cell types or on unusual or aberrant features. And some examples of these include fibromyocytes, SEM, or fibrochondrocytes. So we want to know how these CAD risk loci that have been linked to this disease affect cell state changes in the vasculature, very specifically and concretely. So to do this, we look particularly at 9p21.3. This was the first locus in the human genome ever to be linked to coronary artery disease, initially identified in multiple GWAS studies in 2007. And it is, to this day, the most impactful single genetic risk factor for coronary artery disease. And it's a pretty interesting region to have such a significant impact because it is not only a non-coding region, it's actually a gene desert, so it doesn't contain any genes at all. What it does have are around 80 single nucleotide polymorphisms in very, very strong linkage disequilibrium in most populations. So they inherit together and they form a haplotype that has these two alleles, the risk allele, which confers this increased risk of CAD, and the non-risk allele, which does not. It's also worth noting that this region, although it doesn't contain any genes, it does overlap a long non-coding RNA called ANRIL. And in addition to this, there's antral on the diagram. You can see how much it overlaps with the region. And this region is also only conserved in higher order primates. And this has really influenced previous work that's been done into 9p21. So this region was identified close to 20 years ago now, and there's been lots and lots of excellent research trying to parse out exactly the impact of the region, including attempts to delete a putative ortholog region in mice. It is worth noting that this ortholog does not have the 80 single nucleotide polymorphisms that define the risk from the non-risk allele in humans, and nor do they have antral. But people have attempted to study it, and deletion of this region has uncovered a connection to proliferation in smooth muscle cells of the aorta, but very notably no connection to atherosclerosis. So there's, of course, questions around how to interpret this and how to apply it to human disease and human studies. Another complication of having it conserved only in humans is that we, of course, cannot genetically edit people. So that has really limited previous studies, and specifically in their ability to draw a cause-effect relationship between genotype and phenotype, which is difficult to do without being able to genetically edit. So we address this by using iPSCs, or induced pluripotent stem cells. And this allows us to take peripheral blood mononuclear cells from a donor, human donor, with the genetic background of interest and somatically reprogram those cells into stem cells. And this system has a variety of benefits. Of course, once we have stem cells, we can make any type of cell that we want. In our case, we want VSMCs, but we could eventually do co-cultures with other cell types or just start looking at other cell types. It allows us to maintain the genetic background of interest, like the 9p21 allele at the risk locus, and it also, the somatic reprogramming erases epigenetic marks, effectively minimizing any influence from the patient's lifestyle or previous medical interventions. And finally, most critically, we can genetically edit these cells, and this allows us to establish a cause-effect relationship between the genotype, the 9p21 variant, and whatever phenotype that we see. So in 2018, we used this model to publish a study that looked at two donors, one who was homozygous risk for the 9p21 locus and one that was homozygous non-risk. And we went through the process I described, taking their blood, somatically reprogramming the cells, and obtaining induced pluripotent stem cells from these donors. And once we had those iPSCs, we then genetically edited them to completely knock out the 9p21 region, which I've marked again on the diagram just here. So that generated our four genotypes that we work with: the risk intact cells, the isogenic knockouts that are genetically identical, just missing the 9p21 region, and then the same in the non-risk background. And once we had those four genotypes, we differentiated the cells into vascular smooth muscle cells, and we started looking at the differences to see if we could uncover the impact of the 9p21 region. And pretty much immediately, we saw a really significant transcriptomic difference being mediated by 9p21. So we saw that there were more than 3,000 genes that were being differentially regulated in the presence of the risk haplotype. And really critically, we noticed that when we knocked out the 9p21 region in the risk cells, this transcriptomic profile was essentially reverted back to the non-risk. You can kind of see how the risk cells here in red are very physically far away from the other three groups on this figure here. And this told us that it really is 9p21 in a causal effect, single-handedly driving this transcriptomic difference. Now, once we started digging into those transcriptomic differences, we saw genes related to things like cell adhesion and cell proliferation. And when we looked at functions of these cells, we saw corresponding functional defects, with our risk cells exhibiting increased proliferation, reduced adhesion, and reduced contraction. Finally, we also checked on this long non-coding RNA that overlapped the region, antral. And we saw that there was an enrichment for this gene in our risk cells. You can see it starts increasing in the risk cells, this red line, on day 5, and the difference only gets greater as they head towards day 17 and mature vascular smooth muscle cell identity. We also found that overexpressing this gene in non-risk cells could partially recapitulate the transcriptomic differences we saw in the risk cells and reproduce the adhesion defect we saw in the risk cells, suggesting to us that this long non-coding RNA is playing a significant role in the 9p21.3 mechanism. So coming out of that work, we wanted to know if 9p21 is driving a VSMC cell state that resembles one of these previously reported disease states for VSMCs during coronary artery disease, and if so, by what genetic mechanism is it exerting this effect? So that brings us to the work that we just published recently, where we took our same four genotypes, the risk intact, the isogenic knockout, and then the same in the non-risk background, only this time we sent them for single cell sequencing to see if we could dig more deeply into the differences between the genotypes. So here we have that data on the left. This column is our iPSC-derived vascular smooth muscle cell single cell RNA sequencing data. And on the right, I have a single cell RNA sequencing data set from this publication, Hu et al., which was looking at healthy human heart arteries. And you can see I have 3 smooth muscle cell markers here, and we're comparing how these smooth muscle cell markers express in our iPSC-derived VSMCs to how they express in the smooth muscle cells from the endogenous human arteries. And there is quite a strong overlap in how these markers are expressing. So this validated to us that our model is some reflective of something that you would see in an actual human artery and not just kind of a series of artifacts from culture conditions. But to further validate this, we collaborated with Dr. Clint Miller's lab over at the University of Virginia. They have this lovely metaplaque atlas that is comprised of multiple single cell data sets from endogenous human arteries, including samples from both atherosclerotic and healthy donors. And we asked them to project our cells into this atlas to see where our iPSC-derived VSMCs were overlapping. So our cells are pictured here in gray, and then the endogenous smooth muscle cells are this pink population in the center of the UMAP. And you can see there's quite a significant overlap between our iPSC-derived vascular smooth muscle cells and the endogenous smooth muscle cells. Once again, validating to us that our system is reflective of endogenous VSMCs and something that's useful to study human disease. So having ascertained this, we started digging more deeply into these single cell data that we generated. And one of the first things we saw was that there was quite a bit of heterogeneity within our vascular smooth muscle cells in general. We saw 10 clusters when we did clustering analysis. And this was another positive indicator to us, since VSMCs are known to be quite heterogeneous in vivo. And this indicated to us that our model is something that's able to capture the variability that is kind of inherent to this cell type. But we also wanted to see how our genotypes were grouping within this UMAP. So here on the right, I have the same UMAP colored by genotype, and you can see we have the 2 non-risk lines in green, and then the risk knockout line in yellow. And you can see that all 3 are kind of intermixing in these 3 portions of the UMAP. And this is in strong contrast to the pattern we see when we look at our risk cells, which are all off by themselves in this upper right portion of the UMAP. So this, similar to what we'd seen previously with the risk cells exhibiting a unique transcriptomic profile, and the non-risk or the risk knockout cells essentially being indistinguishable from our non-risk cells. We also wanted to look at where our clusters were grouping with respect to these genotypes. So we made this bar chart and we identified these 3 clusters, clusters 3, 4, and 6, as containing the majority of our risk cells and very, very few cells of other genotypes. So these were our risk clusters moving forward in the analysis. So having seen these subpopulations in the risk cells, we wanted to see what kinds of cellular processes are defining these clusters. So we did a gene ontology enrichment analysis, and you can see that these are the 3 risk clusters, clusters 3, 4, and 6. And I've separated them by the terms associated with upregulated genes and the gene ontology terms associated with the downregulated genes. And pretty much right off the bat, we saw a pretty interesting pattern emerge with a bunch of terms that you would expect to see upregulated in healthy, normal vascular smooth muscle cells being downregulated in our risk cells. So things like cell substrate adhesion, wound healing, these are normal VSMC functions, but here they're being downregulated. And then in the upregulated terms, we saw quite a few gene ontology terms that are not typical at all of normal, healthy vascular smooth muscle cells. So things like cartilage development, chondrocyte differentiation, and cluster 6, which had all of these neuronal developmental terms. So this indicated to us quite strongly that although our risk vascular smooth muscle cells do continue to express smooth muscle cell markers, they are not behaving normally. They are aberrant in some way. To further confirm this, we did a pseudotime analysis on each of our genotypes separately. And you can see here, we anchored the pseudotime in cluster 2, which contains around even amounts of all of our genotypes. And all the trajectories proceeded from cluster 2 towards the right of the UMAP. However, the three not risk genotypes, the non-risk and the risk knockout cells, they all went down as they proceeded towards the right of the UMAP, with the non-risk lines terminating here in cluster 0 and the two knockout lines terminating here right next to it in cluster 7. Whereas when we look at the risk trajectory, as it proceeds from the left to the right of the UMAP, it moves upwards, terminating up here in cluster 4. Once again, suggesting to us that the risk cells are diverging from the other three genotypes, although they continue to express smooth muscle cell markers, excuse me. We also checked on the transcription factors that were driving each of these trajectories, and we saw that there were two transcription factors that were common and unique to the risk knockout and the non-risk knockout. So the two knockout lines, SOX4 and FOXM1. We saw that there was a group of transcription factors that were common to both non-risk cell groups and the risk knockout, and those are colored in green. And finally, we saw quite a few transcription factors that were completely unique to the risk trajectory, indicating to us that these trajectories are being driven by different modules of transcription factors. So having established that the risk cells have taken on this aberrant or divergent cell identity, we wanted to see if we could see any similarities between this risk cell state and any of the previously identified VSMC disease states in coronary artery disease. So one of the first things we saw was an enrichment for this transcription factor, SOX9, which is associated with osteoblast or chondrocyte development or bone or cartilage cells. And this is also something that we saw represented in the gene ontology enrichment analysis a few slides ago, if you recall. And when we dug deeper, we found also an enrichment for collagen 2 concomitant with a decrease in collagen 1, which you can see here on the violin plot. And these are all indicators of an osteochondrogenic signature. Finally, we also checked for actual markers of osteoblasts and chondrocytes. And sure enough, again, we saw an enrichment in the risk cells relative to their isogenic knockouts, all of which really strongly suggested to us that our cells are taking on an osteochondrogenic-like state. Excuse me. And this is something that we have seen reported in the literature. So I have here some figures from the Miller Labs 2023 publication with that wonderful Metaplaque Atlas. And they did this analysis where they took their smooth muscle cells and isolated them and did a trajectory analysis with just those SMCs. And what they found was that the trajectory proceeded from contractile smooth muscle cells up through transitional smooth muscle cells. And then there were two possible cell fates, the fibromyocytes and the fibrochondrocytes. And these fibrochondrocytes are vascular smooth muscle cells that are exhibiting a modulated state in which they've taken on osteochondrogenic-like features. So we asked them to project our iPSC-derived VSMCs into this pseudotime trajectory to see where our cells were overlapping along the transition from contractile to fibrochondrocyte. So here is the initial integration of our smooth muscle cells with their Atlas smooth muscle cells, and our iPSC-derived cells are in gray, and the fibroblasts were included for context. And here it is again, but annotated for some subpopulations within the cells. And already you can kind of start to see our iPSC-derived cells are overlapping with this orangey, peachy population. Those are transitional smooth muscle cells, as well as having this long projection headed downward on the UMAP. And this is the pseudotime analysis that they did with this integration of our iPSCs with their smooth muscle cells from their plaque atlas. And what we see is, once again, this transition from contractile smooth muscle cells down through transitional smooth muscle cells to fibrochondrocytes and fibromyocytes. But what becomes clear here is that this population of cells that's kind of sticking out of the bottom of the UMAP, it's proceeding along a parallel trajectory to the fibrochondrocytes. And when we look at the genetic breakdown or the genotype breakdown of that portion of the UMAP, you can see that it's really overwhelmingly majority risk cells. So what this suggested to us is that our risk cells do resemble fibrochondrocytes seen in endogenous human arteries, but they appear to take on an even more exaggerated version of the fibrochondrocyte phenotype. So having established this osteochondrogenic signature in our risk cells, we wanted to see how it was impacting the actual function of the vascular smooth muscle cells. So we did a calcification assay, and this involved not only staining for calcium deposits with alizarin red, but also subjecting the cells to treatment that encouraged calcification to see if the different genotypes responded differently. And you can see in these images comparing risk cells to risk knockout cells. Even at baseline, the risk cells are showing higher levels of calcium deposits. And when we treat them with the calcification media, the difference becomes really quite extreme. And you can see it quantified over here in this bar chart, showing that the risk has an overall greater level of calcification, no matter if we treat it with calcification media or not. But it also shows a stronger response to the calcification media than the risk knockout cells do. And then down here, we have additional assays that we did with 2 additional risk donors and some non-risk and non-risk knockout cells to confirm that this is a phenotype that we continue to see across donors and across genotypes. And the pattern did indeed hold, with the risk cells overwhelmingly exhibiting greater calcification than any other genotype. Now, this is something that is precedented in the literature. 9p21 has been linked to calcification. I have here a nice GWAS study looking at variants linked to calcification, as well as some figures from this paper, Kojima et al., that looked at atherosclerotic lesions from humans and did find greater calcification in individuals who were carrying the risk allele at the 9p21 locus, as well as higher rates of plaque rupture in those individuals. We also did a migration assay, since a decreased migration is associated with the osteochondrogenic phenotype as well. And what this involved was seeding a layer of cells and putting a scratch down the middle of the cell layer, and then seeing how long it took for the cells to fill in that wound. So this top row of images on the slide, these were taken on the day the scratch was administered. And then on the bottom is images taken 4 days later. And you can see the risk knockout line and the 2 non-risk lines, they've pretty much completely closed up the wound by the 4th day. But when you look at the risk cells, they've barely made any progress in closing the wound that was made, exhibiting greatly decreased migration capabilities relative to the other genotypes, which you can see quantified in these figures down here. These figures, it's also worth noting, do include an additional risk donor and additional non-risk donor to once again confirm that this is a phenotype that we see across donors and not just variability between individuals. We further confirmed this phenotype in some primary samples that we had. And in this case, we were looking at primary samples taken from an individual who was homozygous non-risk for the 9p21 locus and an individual who was heterozygous. And here we not only see that this phenotype is consistent in primary samples, we also see that we still see an effect from a single copy of the 9p21 risk allele in this heterozygous individual who still shows significantly decreased migration capabilities. So having established that we have this very distinct osteochondrogenic signature in our risk cells and that there is a significant impact on the cell's function that's coming from this osteochondrogenic signature, we also wanted to see whether we could identify any genes that potentially haven't necessarily been reported in connection with vascular smooth muscle cell modulation and coronary artery disease. So we returned to our single cell data set and we found these two candidates, LIMCH1 and CRABP1. And you can see here, these are figures from our single cell data set that show that they are enriched in our risk cells. And then we have qPCR results with additional donors for both risk and non-risk, once again confirming that this is a pattern that we continue to see across donors. And we further confirmed this with some immunocytochemistry. You can see here we're comparing risk to non-risk. We did an additional analysis looking at how these genes were expressing together in our cells. And what we saw was this pattern here with all these different purple portions of the bar chart being LIMCH1 positive, but in different combinations with CRABP1 and with SOX9, one of our osteochondrogenic signature genes. And what we saw was that it appears that the LIMCH1 gene defines the risk state in together with these other genes in different combinations. So then we wanted to see what ANRIL has to do with all of this that we've been seeing about the osteochondrogenic signature and these two novel genes that we've identified. If you recall, ANRIL is this long non-coding RNA that overlaps the coronary artery disease risk region. And earlier, I showed you all our previous findings that there is an enrichment for this gene in our risk cells. This enrichment that we see is actually an isoform-specific enrichment. So long non-coding RNA ANRIL is a heavily spliced gene with many, many isoforms. And here in this diagram on the top right of the slide, the isoforms that show up in our iPSC-derived vascular smooth muscle cells are indicated in dark gray. And you can see there are 4. And 2 of them end over here in exon 19. We call these ANRIL long isoforms, and 2 of them end here in exon 13, and we call these ANRIL short isoforms. And what we were seeing was that it was the short isoforms that were enriched in the risk cells specifically. And these isoforms were the ones that were able to induce the adhesion defect that we saw in the risk genes and partially recapitulate the transcriptomic differences we saw in the risk cells. So we overexpressed these short isoforms of ANRIL on some non-risk knockout cells to see if it was impacting these osteochondrogenic signature genes or these novel risk signature genes that we identified. And sure enough, both SOX9 and collagen 2 osteochondrogenic signature genes and LIMCH1 and CRABP1, these 2 novel genes, all of them responded to the overexpression of the short isoforms of ANRIL, indicating to us that ANRIL is a major driver of this osteochondrogenic state that we are seeing. Okay, so that brings me to the conclusions of our paper. What we concluded was that the 9p21 CAD risk locus promotes the transition of iPSC-derived VSMCs towards an osteochondrogenic phenotype. That iPSC VSMCs carrying the risk haplotype display a distinct transcriptomic signature characterized by these osteochondrogenic features and these novel markers LIMCH1 and CRABP1. We saw that the knockout of this entire haplotype reverts this signature essentially back to a non-risk state, establishing a really strong cause-effect relationship between the 9p21 risk locus and all these transcriptomic and functional changes that we're seeing. And we saw that the short isoforms of the long non-coding RNA ANRIL are sufficient to induce this osteochondrogenic transcriptional signature. Finally, we also saw that iPSC-derived VSMCs exhibit a transcriptional profile that strongly resemble those of ex vivo human arterial VSMCs, which show the utility of this model in modeling human disease. And we demonstrated that this 9p21.3 driven cell state transition is a cell autonomous process that occurs even in the absence of any cues from the microenvironment that you would find in a native human artery. So that was a lot, but I also wanted to briefly discuss the impact of the review process on this paper. It changed quite a bit from the very first draft we submitted to the one that ended up being published. And one of the issues that came up was this issue of inter-individual variability and the question of how many donors we're using and where we're using them. You might have caught how often I repeated that we validated results in additional donors throughout this presentation. And you can see here kind of a diagram of how that went. We had our single cell data set that had only the 2 donors where we did our analysis, and then we confirmed results with additional donors and with primary cells. And this was something that changed as during the review process, both in that we added more assays with additional donors as time went on and it became clear that we needed more, but it also changed in how we discussed it in the paper. Because one of the things that we ran into quite a bit is that it was clear that it was difficult for readers to pick up when and where we were using these additional donors. And some reviewers were being left with the impression that we weren't confirming our results in additional donors, and they were very worried that we were just kind of picking up differences between the 2 individual donors from the single cell data set. So one of the changes we made, aside from adding additional donors throughout the process, was also we became very careful about how we refer to them. So I have here 2 versions of this figure from the calcification assay, and you can see the way we've labeled them has changed, and we've also added this non-risk cell group to the figure. We also ran into issues with batch correction. So all of our cells were cultured simultaneously and in parallel. So we kind of went into this thinking that we didn't really need to do a batch correction. But we did end up doing one just to really confirm that even with harmony corrected data, we still see the same pattern, the same 10 clusters in the single cell data set, and the risk cells kind of clustering by themselves in this portion of the UMAP on the right. And one last one that I'll bring up is the addition of this calcification assay, which is not in the first draft of the paper. We also heard some concerns from people who were looking at our research about functional readouts to confirm the impact of the osteochondrogenic transcriptomic signature that we were seeing. So this was an assay that we added kind of later on in the review process, thanks to a lot of hard work from one of our undergraduates, actually, who developed this assay for us. And with that, I would like to thank all of my funding sources, as well as the members of my lab for all their support and contributions to this paper. I'd like to thank my mentor, Dr. Losardo, for her endless patience in this whole process. And of course, our collaborators at the University of Virginia, Dr. Clint Miller, and his graduate student, Jose, who did a lot of the transcriptomic analysis that we saw in the integrations with our data and the metaplaque analysis. Thank you.
Great. Thank you so much, Elsa, for this wonderful presentation. And I'm really happy, coming from the smooth muscle cell field, to hear that there's more genetic evidence coming up linking some of the GWAS hits actually to smooth muscle cells and then CAD. So this is really good and keeps us in the business, I guess. But I will leave the floor now for questions. So you can feel free to unmute yourself and ask a question, or also you can type it in the chat.
Melody, I just wanted to also say that I saw Clint is actually on the call as well. I'm very happy that
Hello,
he's also here. So thank you, Clint, for participating.
Thanks for
And
joining.
like endless thank you to his group really for doing this beautiful analysis with the ex vivo samples. So Clint is here for the hard question. Just ask him.
Perfect.
Yeah, thanks for.
Hello.
Yeah, it's great to join in, and it's a beautiful presentation, Elsa. Really nice work. Jose is out of town, but he would have joined as well. So
Yeah, thank you all
Thank
for
you all
joining.
for
Like,
joining.
we are so happy that you guys accepted to share your work with us, and beautifully presented as well, Elsa. There was a very nice presentation and overview. I think that was super clear. And if any shy audience member has a question, just please come ahead. I think I just wanted to maybe start with a comment while people find the courage to ask those million questions that we all have. Um, I would probably start from your final slide. So the bioinformatic part is super interesting and so powerful here, but probably for the young crowd, what would be your comment about the difficulties to uh, corroborating in vitro through like functional assays the findings that we often times see at the bioinformatic level, because the findings here are so powerful. But I would like your opinion in around like how important was for you to find the right functional assay, I would say, and to support further your findings.
Yeah, definitely. So it was pretty critical, I think, to find the right functional assays to support the case that we were making. Because for a long time, the work that I was doing was almost purely computational. I was really focused on the single cell data set, and we were finding lots of interesting things. But even from my professors in my committee, we were hearing that we needed to confirm something in terms of the functions of the cells. Uh, so we did have the osteochondrogenic signature to go on to kind of try to find what the right functional readout was. And we got very lucky with the calcification assay. It was pretty easy to do. And we had these lovely, very distinct results with the differences in calcification between the risk and the non-risk cells. The migration assay followed kind of a similar path. We started looking into it because we were seeing some differences in the expression of metalloproteases between the risk cells and the other genotypes, which are, of course, associated with migration. So that's kind of how we started looking into the migration assay. And it was really kind of inferring from the transcriptome what may be happening in the cells and then trying assays to see what worked. And of course, we ended up with some very lovely, very distinct phenotypes when we looked into that.
If I can add something, I feel like, the migration assay was something that we were doing sort of also in parallel. There was another technician in the lab that was looking at migration assay. We had this, I guess the behind the scene is that we had this very fancy new instrument in the lab. So there is just a microscope in the incubator, basically, and you can do a bunch of assays with that in sort of real time and live imaging. So he was trying a lot of these different assays, doing migration with different cells in the lab. And we found this very strong phenotype. I think that, that came together very nicely with our single cell data set. We had also a lot of gene ontology terms that were indicating that we had a defect in migration, or there were a lot of genes that were involved in migration that were downregulated in our risk cells. So in the meantime, Rowan was doing all of these migration assays, and we saw that the cells were actually really impaired in the migration process. So that, fit very nicely with the GO-term analysis. And then, looking also in the literature for fibrochondrocyte, mostly functional properties, we saw that there were evidence actually showing that fibrochondrocyte in the atherosclerotic plaque are not actually, migrating. So that was, I think, a lot of different pieces put together, not necessarily, consequential. We didn't go really, one after the other. So, while Elsa was working on something, other people in the lab were actually focusing on different projects and end up being very nice story put together.
It's always a team effort, right? Isn't it?
Can I jump in here and ask
Go,
maybe
Melody.
what are your thoughts about the specific osteochondrogenic SMC phenotype when it comes to clinical outcomes? Do you think on the long term, is it something that we want more to have in the plaque? We want less to have in the plaque? Is it a good thing? Is it a bad thing? Where do you stand on this? Where do you think it stands on the spectrum of all of the different SMC phenotypes that we find the plaque?
I mean, right now it's looking like it's a deleterious phenotype for sure. The figure I showed you in the Kojima paper that found more more calcification in the atherosclerotic plaques of individuals with the 9p21 risk allele also found that these individuals had a higher incidence of plaque rupture, which is, of course, one of the ways that coronary artery disease becomes deadly. It causes a thrombosis. We also, with the reduced migration in our cells, we've been speculating with this idea that it might prevent the vascular smooth muscle cells from forming the fibrous cap that goes over an atherosclerotic plaque and help stabilize it and prevent it from rupturing and causing a thrombosis. We think maybe the reduced migratory capabilities make the cells less able to form that cap, which would, of course, be deleterious and make the disease deadlier. So right now we're leaning towards this is not a protective phenotype and is not something we want to see in the disease.
Very cool. I would follow up just with another note. I think in one of the first slides that you showed about the locus specifically, I think one of the genes that I also saw was I think it was the senescent associated gene, which was CDK2NA. I think it was also close to this locus, and I know also from the cardiogram consortium studies that it's also a very high CAD hit as well. So I don't know if there's some sort of interaction between this locus and then the part that codes for the senescent related. I'm not sure if it's P16 or P21, but it's also a good, maybe this locus in general and maybe what's around it are very much related to CAD. So just caught my attention. Do you know anything about that, or have you explored that?
So those are the two closest protein coding genes to the region. Previous research has been kind of inconclusive in connection with a 9p21 specifically. It kind of depends on what tissue type you're looking at, even what like SNPs within the region. Different studies have kind of found different results. And in our data sets specifically, we didn't see a tremendously compelling reason to be looking into them with our iPSC-derived VSMCs. But definitely it's something that is still kind of a question mark for sure.
Thank you.
I think that, like we actually added in the paper, there is a supplementary figure that we added about, the expression, not only the CDKN2A and B, but also all the other genes that are within this, region, even farther, downstream of the 9p21 CAD locus. I think we see some gene expression differences in the CDKN2B. And but I don't think they don't match consistently with all the rest of the signature, and so we don't think that those are main driver of the signature. There is definitely, we've seen even previously in our previous studies, several years ago, that the risk cells are more proliferative. They definitely proliferate more. So there is an effect of the CDKN2B gene. I think we see a downregulation in the risk cells. The cells proliferate more, but also our non-risk knockout are super proliferative as well. And the difference in just the CDKN2A and B doesn't seem to explain everything that we see, down the line. So I think that there is an effect definitely on proliferation, but I don't think that it's only that. Or I guess we don't know how to, in the past, we have tried to modulate the CDKN2B expression. But it's very hard to do because, as you said, it's a gene that, induces actually senescent. And so like the right amount of these gene to like, promote proliferation or promote senescence, it's very hard to modulate. So we tried at one point, not really successfully, I would say, even when I was a postdoc, and then we sort of, gave up on that.
Very nice.
That's a really good
question.
point.
Oh, sorry.
Just to follow up on the senescence, it may not be smooth muscle cell specific.
Right.
The previous report that linked 9p21 to senescence was related to endothelial cells, I think.
Right. Okay.
But there's got to be some link, as you mentioned, and maybe the chromatin interaction studies, where you can knock out, given variant or region, and then look at how that impacts chromatin structure with the neighboring genes. That could be interesting. But
Definitely.
yeah, definitely there has to be a link, and it may not be specific to smooth muscle cells.
Maybe more complicated than we think.
Yeah, yeah, it always
Very
is,
complex.
Melody, right?
Thank
So
you,
I
Paul.
think we may have a question from Miran. Do you want to come in, Miran?
Yes, thank you. Can you hear me?
Yes.
Yes.
I'm continuing my tradition of tuning in with from the obscure locations. Today I'm on a parking lot in my car in front of my building. Yeah, in any case, thank you so much. This was such a phenomenal work. I read this paper a few times. We also kind of went through it on our internal journal club, and now it was kind of joy to see again all the details behind it. I have a two question. One is more maybe speculative because your study was mostly focused on coronary artery disease. Do you think that this is just a specific thing for vascular smooth muscle cells to produce this phenotype in coronary arteries, or it can be also seen maybe in another types of plaque influenced blood vessels? Because Mel, correct me if I'm wrong, but we didn't observe previously in another, for example, from the carotid plaques, from plaques coming from carotid arteries, we didn't observe similar phenotypes so far. So that's the first question. And the second question is related to the fact that Unreal is really one of the not few, unfortunately, long non-coding RNAs that have many isoforms. And you nicely pointed this out, that it's the short one that is really important in this scenario. However, from the technical point of view, it is really very, very problematic to quantify different isoforms because there is overlap between many exons. So if you just can clarify, how did you approach this? I saw that you wrote in the paper that you separately quantify the long isoform and the short one, but you cannot actually quantify the short one. So what was the approach just from the perspective of qPCR quantification? And thank you so much for joining us and presenting this work. Really, really congratulations.
Yeah, thank you. I'll start with the second question. And I pulled up the slides so that you can see the different isoforms here. Basically, we chose primers that picked on specific exons to make sure that we were looking at the isoforms that we were interested in. So you can see like 13 is only showing up in our short isoforms. But definitely it's been problematic to try to quantify the different types of the different isoforms of the gene, particularly as we've been trying to look into it with like bulk RNA sequencing and other kinds of transcriptomic analysis. It's kind of an ongoing problem for sure. But we're pretty confident in our ability to do these qPCR assays and to do isoform specific overexpression in our cells. And those are definitely tools we're employing to kind of get around the difficulties that we're having from the sequencing end of things with our bulk data sets.
And I think, Myron, if I can interrupt also 1 second. So you're right. So there is a lot of overlapping. And then, we basically followed in this paper the same approach by qPCR that we did previously. And this is actually measuring if you see here the schematic that Elsa has, we measure like the exon junction from 6 and 7. And here in this schematic, you can see the darker isoforms are the only one that we actually detected in our smooth muscle cells. So of all of these 17 linear, and actually also the circular at that time, we couldn't find really any other than these 4. So we had 2 long and 2 short isoforms. So we picked exon 6 and 7 junction, which you can see it's in all of them, in all 4. And for us, it's a sort of like, overall, like a total amount of embryos. And then we measured also exon 18, 19, which is only in the long isoform. So in the long isoforms, 18, 19, we don't see any differences in expression. And when you measure basically the total amount, that it's way more. So it's a sort of by subtraction that we say, if the embryo long isoforms are the same expression, but the overall amount is more for the risk cells, these must be the short isoforms. In the past, we confirmed that to, with primers that were picking only in exon 13, which is this exon that only it's present only in the short isoforms, and we see the same pattern. So it's sort of a mix. We usually run multiple primer pairs and multiple junctions to figure out which type of isoform we have. If I can, like just say something about your first question, and I don't know if you're actually hearing me. Can you hear me?
Yeah,
Okay.
I can hear you. Thank you. I
For
understand the
your
first part.
for your first question, I think honestly, I don't think that this phenotype on fibrochondrocyte, it's something that could be only in coronary artery disease. I think that the 9p21 has been associated with abdominal aortic aneurysm, intracranial aneurysms, and other vascular disease. So I think that maybe this is something that could be potentially a sort of applicable, something you can find also in other vascular disease. I have to say, we start now understanding what the different vascular bed signature is. It's true that each single artery or like in different location, they are different. So we assume that all the arteries are the same, but in reality, this is not the case, and we know that. And I think that probably in the aneurysm field, that is way more. I think that is way more studied, or people focus on that way more because why would you have, for example, abdominal aortic aneurysm or thoracic aortic aneurysm? Like why would that happen in different portion of the aorta, right? And I think that this is probably the case also on genetic variants that are affecting specific vascular bed or maybe multiple like different ones. In our case, I would say our cells seem to match pretty well with, in general, heart arteries. In vitro, we do a differentiation that is usually going through the lateral mesoderm, which is supposed to mimic more maybe coronary arteries. But I have to be honest, of course, these are cells that we make in vitro. And as much as they resemble, like human smooth muscle cells, I'm not sure how much the fine tuning, it's applicable to this system. But we were like amazed actually when Clint was able to do this analysis and, like see our cells projecting into smooth muscle cells from actual, human arteries. It was really impressive.
Thank you so much. Thank you so much.
Thank you so much for the comment. I think we have another question from Sofia Vichkovic. Would you like to unmute yourself?
Yes, hi. Thank you so much. I hope the echo in the space that I'm in is not so bad. This was a fantastic presentation. Really, congratulations on the very impressive work. And maybe to continue on the discussion that we were just having, I was really impressed by how well the SMCs derived from iPSCs map onto all the different data sets that you have shown. Did you include any specific maturation protocols, or was it challenging to get them into a state where they really do resemble the true SMC? Because we know that in some other cell types being derived from stem cells, this is sometimes a challenge. So can you shed a bit more light on that?
Yeah, so VSMCs don't necessarily have kind of a bright line where we can say conclusively this is the line where we know they're mature VSMCs. Like I have some friends in an islet lab, and they have a nice line because they can say once the beta cells start producing insulin, then they're mature. We don't really have that. But we do have markers for vascular smooth muscle cells that we can check to determine whether they've taken on that identity correctly. But I also was fortunate enough to come to the lab after we had developed our differentiation protocol. So Valentina probably has more comments about how exactly it is that we came to that protocol.
I think that, this is a protocol that it was published before in 2014 from the SINA lab. So that has been working actually very nicely with us. One of the challenges with the iPS cells is just that you can differentiate cells in any cell type. Most of the protocol will give you a good percentage of purity or homogeneity. Most of them actually not. So we were able to replicate this protocol to use it in our lab to have almost 100% sort of conversion of the cells into smooth muscle. I think, we strictly follow the protocol. We added in time a couple of changes, but not that many. And the cells at the end, after actually about 15 days of differentiation, 17 days, they start expressing they expressed already all the markers that we check for, alpha smooth muscle, actin, tagulin, CNN. I have to say our cells in vitro did not express myosin heavy chain 11, which is something that they don't match. For example, in cleaned projection and pseudotime analysis, they don't project with contractile VSMC, which is something that people have seen this in vitro, even with primary culture, right? I think that this is probably one of the limitation of the in vitro culture. So we make cells that are not, they are contractile if you measure contraction, but they don't have a lot of expression of myosin heavy chain. So this is something that, we have always seen it. I'm not sure how to explain that. I feel like you can find the expression of this marker just if you take out the cells directly from a mouse of a tissue and you single cell. I think that the Atlas, the Metaplaque Atlas has, cells in contractile state. They have expression of more mature marker. But we don't add anything other than, following the protocol. So we never try to push the cells even farther. We have tried to do some, maybe some treatment with TGF beta or PDGF, but it doesn't change that much, honestly, in our hand. So in the projection, if you see also in the paper, our cells mostly overlap with transitional SMC. So I think overall, they resemble mostly transitional SMCs. And then we have some lineages into fibromyocytes as well. And then we have our risk cells that are more into fibrochondrocyte. I don't know if that answered your question.
It did. Thank you so much.
Thank you for the brilliant discussion. I think we might have time for a quick final question, if there is anyone with a burning one.
I have a curiosity. Sorry, I've been trying to raise my hand, but I don't know how to do it.
Go ahead,
I
please.
couldn't find it. Sorry. No, it's a curiosity. I was just wondering if there is an increased risk of a deleterious phenotype associated to this locus during aging, or if you have some kind of correlation with aged patients, or if you have stratified, I don't know, the different cells that you've been differentiating, the iPS, for example, if they are age specific, or you're using a specific age group of patients, or if there's something known with respect to that.
Yeah, so in the work that we've been doing, our cells are coming from donors who are 50 to 60 years old, is it? Yeah. but we haven't necessarily been doing specifically aging studies trying to see an impact with that. And to the best of my knowledge, I think previous studies in 1921 don't see any particular relationship between age and the locus.
So I think that overall, like most of the cells that we have, especially from risk homozygous risk donor, these are cells that come from both people that actually had myocardial infarction early on in their life, and also people that are totally healthy. So we looked at sort of genotype specific, sort of phenotype, cellular phenotype, independent of like the history of the donor. Some of our, healthy donor are actually very, very old, like in the 90s even. So, but our cohort of sample is too small, I think, for doing like, a good stratification. I think the beauty of this work that we can do with iPS cells is just that it becomes powerful even with a small sample size, right? Because when you do isogenic line that are edited, so you can control for genetic background and you can only see at one specific region you're interested in, it is a small sample size. Like our approach is we do the isogenic line for a small number of sample, and then we use like, additional donor, maybe unedited. At that point, we don't need to do genome editing of everything. You know what I mean? It's just we use additional donor just to look into specific markers. But I think still with our cohort, we're not able to do any sort of aging correlation yet. We need a bigger cohort.
But
Thank
maybe
you.
to follow up on that, it could be possible in these population studies, and maybe if people have looked at coronary disease versus PAD in more age stratified cohorts, and just maybe seeing 9p21 is more strongly associated with individuals greater, let's say 65. So that I think there is a possibility that it could be a greater risk factor in those individuals, but from these studies, yeah, I agree. We need more donors. We need to increase sample size.
Thank you.
Thank you so much for this wonderful discussion, and then for coming to AtheroNet and presenting your work. I hope that you can join more of our journal clubs later on, now that I'll add you to the mailing list. We also have some career events as well. So if there's any early career researchers that would like to know more about, like next time we're going to talk about we're going to have an editor from cardiovascular research to give us some tips about how to get our papers up to standard, and then also about spin-off companies from students that left academia and then joined another path. So join us, please,